Welcome, everyone. I'm Shelley Frisbee, the CFO, COO of Cancer Commons, and want to thank you for attending today's webinar entitled Testing Matters, Understanding Cancer Biomarker Testing and How It Applies to You. For those of you who are new to Cancer Commons, we are a nonprofit organization that provides compassionate, personalized support and education to advance cancer patients and their loved ones. We help our clients understand and access diagnostic and treatment options so they can be confident they are making the best possible decisions. Cancer Commons is a team that is here for cancer patients and their loved ones. Just a few logistical things to mention before we start. Our PhD scientists will be answering questions at the end of the webinar. Please feel free to submit the questions at any time via the Zoom question and answer feature. Your questions will not be seen by other attendees, and you have the option to submit them anonymously. A recording of the webinar will be made available to all registrants. Thank you so much for joining us today. And now it is my pleasure to introduce Cancer Commons scientists, Dr. Kaumudi, Bhawe, and Dr. Adrienne Nugent.
I'm Dr. Kaumudi Bhawe.
And I'm Dr. Adrienne Nugent.
And we're here today to talk with you about cancer biomarker testing and why it's so important to know about. But before we begin, both of us are going to give a small introduction about ourselves. So I'll begin. I have a PhD in public health. And prior to that, I've worked in industry and academia in preclinical oncology, clinical oncology, early drug discovery. I've basically have work experience leading projects in areas all across from right where you identify targets that could be interesting in cancer, all the way up to now talking with patients about what are the different options that they have and what are the possibilities that they can discuss with their oncology team as I work here at Cancer Commons. And Adrienne, handing it over to you.
Sure, thanks, Kaumudi. So I have a PhD in genetics and genomics from Duke University. I've worked in the academic, industry, government, and nonprofit sectors. So I have got to explore the role of biomarkers in diagnostics and therapeutics in all those different settings. I have worked in the wet lab, kind of really developing these techniques and doing the nitty-gritty work like of next generation sequencing and things we'll talk about today. And then I've been more in the dry lab doing the bioinformatic data analysis and translating the information and results to patients and physicians to help best improve their care. So look forward to talking about this.
Perfect, thank you. And the reason both of us are here is because both of us had some some rude introduction to this very devastating disease as children. And that's what drove both of us into this field. And so we are both very passionate about this, and we are with you. And we want to make this sort of an open discussion, after which we'll have some time for questions. So as well. So yeah, what is cancer biomarker testing? Typically, how does cancer even get diagnosed, right? Typically when someone or their family members are noticing some sort of symptoms, something is off, you go to your local physician or maybe a clinic and get tested for those symptoms. And then maybe there's a blood test, or there's some sort of indicators saying, okay, something's not right. And then the physician will refer you, or in the clinic somehow there's probably an initial scan, which results in a tumor being detected. And that's how, this very devastating diagnosis it happens. And then when you're still digesting that information, maybe you hear from someone saying, oh, you should get biomarker testing done. So, but what is that, right, Adrienne? I mean, we hear this all the time when we talk to patients.
Exactly, Kaumudi. And this field is rapidly exploding. I mean, you'll see there's thousands of new diagnostic testing companies emerging each year.
Right? It's confusing. It's very confusing. So here's some questions that come to mind, right? When you hear those words. First of all, what is it? Even before that, why? Why should I get biomarker testing done? Okay. What is it? When does this happen? When is it important to get it done? Do I get it now? Do I get it tomorrow? When?
Exactly.
And where is it done? Do I get it done at my oncologist's office? Where do I get this done?
And
right?
how do you do it? Is it a biopsy? Is it a blood draw? How is this done? So
Good point.
a lot of questions.
So, first question being, why even do this? Well, sometimes cancer biomarker testing can help in finding the most optimal sort of treatment plan that can be available moving forward. Even at the time of diagnosis, it's something that gets done many times. Maybe it's not even mentioned, but once we look at what biomarkers are, it'll become evident why that this can happen. So just to see what they are, we're using the word biomarker in a very broad sense. So essentially, any sort of marker, either a molecule or a group of molecules that are used to understand the biology of your tumor. So biomarker, that's what that is, and that's what we're testing for.
Exactly, Kaumudi. So this can be a lot of different things. Many of these will bring you back to your high school biology days. There are DNA-based biomarkers, there are RNA-based biomarkers, there are protein biomarkers.
Exactly.
And so all of these can be captured from the tumor or from other body samples, tissue samples, blood samples, depending on what type of cancer you're looking at. So let's talk about when. This is really important. And this is something that Cancer Commons can help individual patients with on their cancer journey, because it's unique to each type of cancer, each patient, when and how often and which types of tests are relevant. And so the big question is, where are you in your journey right now? If you are newly diagnosed, you have the best option for the most amount of data you can collect, right? When you have initial diagnosis, you can work with your treatment team to map out a plan for all the different types of biomarker testing that can be done. So upfront, you have the most information at your hands.
Absolutely. And then at some key decision points as well, this can be done. But Adrienne, you mentioned a really important thing there, that it is also dependent on what kinds of tissue is available. So whether having a biopsy is a possibility or not, whether the blood tests are going to be revealing enough or not. These are things to consider, and that's why having that conversation with your oncology team is very important.
Absolutely. It can be tricky too, Kaumudi. We understand that it's not just the medical oncologist you're talking with. There may be a subspecialist, the surgeon, the radiation therapist. There's a lot of people involved in these discussions. And so it really takes a team effort to understand this and figure out when and where this will take place.
Yeah, and this because it can happen perhaps at the hospital lab itself. This usually many times happens in the bigger cancer centers, but there are also specialty cancer testing labs and companies that do these sort of tests, these different kinds of tests.
Kaumudi, that's a great point. How this is done is rapidly changing as new technologies emerge. So we're going to talk about the current landscape of different ways this can be done. And we stay on top of the emerging data so we understand all the options and tools available to you at time of your treatment. So we at Cancer Commons are trying to stay on top of the latest types of biomarker testing so that we can explain how to access and get these types of tests done. So if you could walk us through kind of the major flavors of biomarker testing. These are pretty standard. And then there's all sorts of little variations that can be tailored to individuals. But if you could walk us through the basic overview, that would be great.
So how is biomarker testing done? As we talked about earlier, typically a tumor or a few tumors are initially found through some scanning. And so any sort of scanning that happens, if it's an MRI, a CT scan, ultrasound, all of those are looking at the tumors from the outside. But really, what are these tumors, right? So if you try to visualize what exactly is happening in the tumor, there are multiple different types of cells participating in making this unwanted, unnatural sort of entity be able to survive and thrive in that environment, right? There are these tumor cells themselves, and then they draw towards themselves blood vessels that are going to nourish the tumor. Those blood vessels are also made of different cells of a different kind. Then there's also the immune cells that are coming from your own immune system. And the tumor is sort of tricking them into helping it remain hidden within the body. So there's this group of different kinds of cells in the tumor. Now, from each of these cells, if you look at each of these cells, the cell itself is quite a complicated structure with in the middle of the cell, there's the nucleus at the center, and that nucleus contains the DNA, the RNA, which has the ability to move from the nucleus into what is known as the cytoplasm, where it's coded into or decoded into proteins. And proteins are the workhorses of the cells that carry out all the different functions. One important take home here is to remember that each of these cells has DNA, has RNA, and has proteins. And luckily, thanks to technology today, the DNA can be isolated or the RNA can be isolated, as well as the protein can be isolated from these cells of different sorts in the tumor. And each of these components can be then analyzed, used for biomarker testing. So these molecules, the DNA molecules, the RNA molecules, or the protein molecules, and various combinations of these molecules form the biomarkers, meaning the markers that give us information about the biology of the tumor that's in the body.
I think that's a really great high-level overview of the different types of biomarkers that we are testing in the clinic today. And now we kind of want to go through each of these one by one. So the DNA, the RNA, and the protein. I'll start with the DNA because this is kind of the traditional starting point. DNA is highly relevant to all areas of cancer. There are a lot of similar terms used in the biomarker field. So we're going to try and define those terms here and also explain synonyms or other words you may hear that have a similar meaning in where you're being treated. So for DNA cancer mutation testing, there's genetic testing, which is referring to your germline mutations. These are mutations that are inherited in your family tree. So here we're showing a family tree, and you can see the individuals with the purple background are representing that they have the same genetic alteration, and they have some sort of trait that's presenting with this alteration. So while we may think of a trait like hair color or eye color passing down through the generations, you can also pass along mutations or alterations that can unfortunately make it much more likely that you will develop cancer during your lifetime. The most common one of this that we think about are the BRCA1 and BRCA2 gene mutations. And these are relatively common and very well studied. And so now we're at the point that when we know people have these mutations, we screen other family members to find out who has these mutations, and they undergo a lot of additional screening and even preventative treatments to prevent cancer from occurring.
Yes. And Adrienne, that's a really good explanation. And this terminology, genetic testing versus cancer genome testing, what is that?
Exactly. So genetic testing, BRCA testing has been around for a few decades. Some of us are quite familiar with this idea. Cancer genome testing has really emerged just in the last decade or so with the ability to sequence a lot of different mutations in the tumor itself. So these are not mutations that are passed down from parents to children.
And and this is important to remember, right? I mean, because it's
exactly right. So these mutations are unfortunately what has caused a tumor to occur in the person, but it does not mean that they are passed down to their offspring. So these mutations we can detect with different types of sequencing assays, which we're going to talk about more. They tell us about the driver alterations, what has caused the cancer. This helps us understand what the prognosis of the cancer, so how likely certain lengths of survival or progression-free survival to certain treatments may be. So these are really helpful. Yes,
exactly. And some things that weren't on this slide I wanted to sort of mention is some terms. For example, NGS. Get your NGS test done. So what does this NGS stand for? So NGS is an acronym or short form for the words next generation sequencing. And next generation sequencing is the technology that's used to do these kind of tests. So that's nothing more than that.
Exactly.
And the next generation sequencing technology is what has brought the cost down of this genomic testing. And that's why it's become so much more widely available now. And it's an option that needs to be considered heavily while making these important decisions in cancer.
As the costs continue to come down, Kaumudi, we're going to be able to explore our entire genome. So could you walk through?
Oh, yeah, Adrienne, yes, thank you for bringing that up. Yeah, thank you. Because some other terms like NGS, some people may say, oh, what about WGS or WES? Okay, so WGS, whole genome sequencing, versus WES, which is whole exome sequencing, versus a gene panel.
Or targeted sequencing, which is just a small subset.
Right.
Could you show us these on?
Absolutely. So we have a pictorial depicting this. When we say whole genome, that means all of the DNA that's present in the cells in the nucleus is sequenced and is looked at for any sort of mutations, any alterations that may exist there. This is typically the most expensive of the different kinds of tests available. This watermelon analogy is just to show you that when you take, instead of taking the whole entire thing, if you take a slice, that would be, for example, the exome. So what is the exome? This is only the part of the DNA that codes for proteins. And so that can be looked at for alterations.
2 to 3% of the genome.
Yeah, exactly. And that's
portion. Yeah,
a very small portion of the genome. And otherwise, in some cases, it's actually more prudent to first start off with a targeted gene panel. In some cases, the gene panels can be large, quite large, with 500 to 800 genes that are known oncology drivers in various cancers, or co-drivers, or just important in cancer cell signaling. Or at times, it can be a very small gene panel, which is looked at first to see if there are any changes there. A few examples of the genes tested for mutations in the different cancers are KRAS, EGFR, BRAF, IDH1. There's a whole list.
Thank you, Kaumudi. That's really helpful. And you can see how it already is very confusing. Do I only need a few genes sequenced? Do I need exome sequencing? Is it worth paying more money for a whole genome, looking for an outside company? Has my provider recommended the proper test?
Along with this genome sequencing, there's some other information that's captured in the tests typically. And we have a pictorial to sort of walk you through that. And then there's three different things you'll see on the report that we're going to show you, or on one section of the report that we're going to show you right here. So here you just see a large blown up portion of a test report showing you 3 different types of biomarkers that are studied. The first one is microsatellite instability. And Adrienne, why don't you walk everyone through this?
Sure. Great, thanks, Kaumudi. Yes, and what you're showing here is that no single piece of information or biomarker testing generally is conclusive. Having a full panel of biomarker testing can really best help inform. So MSI, microsatellite instability, TMB, tumor mutation burden, and LOH, or loss of heterozygosity, all work together to help us better understand the tumor. This, again, is the somatic tumor. So this is the genomic profiling. This is not inherited. Exactly. This is specific to the tumor that has grown in the patient.
Yep.
MSI is a way of determining essentially the stability of the DNA in the tumor. And so this is done by looking at a few different regions of DNA and seeing how relatively stable or unstable the DNA is across the entire tumor landscape. And so when you have stable, a microsatellite instable disease, or microsatellite instability high, or MSI disease, it opens doors to a different class of therapeutics, to immunotherapies that can be very, very effective in patients with this MSI high phenotype. this patient report is showing what's more commonly seen in patients, which is a stable result, which isn't actionable per se, but at least tells us that we've looked for a biomarker that could respond to treatment, and it isn't applicable in this case.
Exactly. Thank you. Yeah, thank you, Adrienne. And then related to this is something called tumor mutational burden, or TMB for short. And this is a little more easy to sort of guess what that is. Tumor mutational burden, meaning that the number of mutations per megabase that you see in the DNA. So a TMB of 10 or more is called a high TMB, and anything below that threshold is called a low TMB. But essentially, this is looking for the number of mutations that are found on the tumor cells. And again, this will give clues as to whether immunotherapies would be effective, predicted to be effective or not for the tumors. Although any one piece of information taken on its own is not enough to give accurate clues about the behavior of the tumors.
Yes, and loss of heterozygosity goes similarly in line to the MSI and TMB. So a way of detecting how much alterations occurred at the DNA level in the tumor cell. And again, if this is high, in this case, if the assay is above 16%, then this opens the door to different treatment options. Exactly. So this again is showing a low result, which would not end the treatment. Okay, Kaumudi, that was really helpful to see the MSI, TMB, and LOH. We now have an alphabet soup of all these different biomarkers. And we're going to continue those acronyms going from DNA to RNA. So the DNA testing, we just walked through and is pretty traditionally done. RNA testing is now more commonly being incorporated into the cancer genomic profiling landscape.
There's two different things you have to remember right now, at least as it stands as of today, is there's something called gene fusion testing, which we'll explain in a minute. And there's another thing called expression-based analysis, which we'll also have a pictorial and talk a little bit about. But these are the two main types of testing, which are RNA-based.
Yes, why don't you start with the newer one, that's the gene fusion testing, like we just mentioned, because this is a next generation sequencing, NGS-based testing, and then I can talk about the gene expression profile.
Sounds great, Adrienne. Sounds great. So the gene fusion testing, a gene fusion is exactly what it sounds like. It's where two parts of different genes have, for some odd reason, decided to come together and work together, form a fusion protein, and start its work in the cancer cells. This is unwanted work, and we're going to show you this slide depicting what's happening here. When we're testing for gene fusions, we're testing for this sort of phenomenon that I just described, where you have one regular gene and another one, and then in the cancer cells, for some reason, a portion of this gene jumps around and fuses with a portion of this other gene. And now you have what is called a gene fusion. This particular gene, NTRK gene fusions do occur in cancers, and there's targeted therapies for this that are being tested right now as we speak. And that's why this example here. So essentially you have this happening at the level of the DNA, and it could be detected by these genomic tests, but it's much easier to detect this at the level of RNA, where the messenger RNA is a hybrid transcript. It's a gene fusion transcript, and it can be detected based on RNA. It could also be detected at the protein level, but we're not going to talk about that. This is the fusion protein, which is expressed in the cancer cell and causes chaos, basically. Some examples of these kind of fusions, which can be tested for, are these NTRK, ROS1, RET fusions, ALK fusions. You know, these are just some.
Exactly. Kaumudi, what's so exciting about these fusions is that they're really amenable to targeted therapies. The way that they form creates a nice drug binding pocket that we can then develop these targeted therapies that are very efficacious. So although these are rare in general in solid tumors, and certain tumor types, they're very common.
Yeah.
And very actionable. So it is worth doing.
Absolutely. It's important to ask your oncology team, does it make sense to get this sort of testing done?
Absolutely.
Or also, does it make sense to get this sort of expression testing, which Adrienne, you're going to talk about now, right?
Yes. And
So.
this all goes back to our very beginning of the conversation, is when and how and why do this. RNA, we haven't touched on yet, Kaumudi, is very fragile. So, and very, takes a lot of resources to get RNA testing done. So it's important, if we had an unlimited amount of sample and time, we would do RNA testing perhaps.
Yeah. Also, another thing is many times these tests, I mean, you really have to find out whether they're covered by insurance or not.
Yes.
So that's something to think about.
Great. So Kaumudi has nicely explained the gene fusion testing using next generation sequencing. We also have the ability to use RNA to test gene expression patterns. And we can do this as well with next generation sequencing. And before that, we used a technology called microarrays to determine gene expression in tumor cells. So this is what's called a heat map. And you can see across the top, each column represents a different individual patient. And going down in rows, there are individual genes represented. And what's been done is the RNA has been taken out of the cell, and the expression level, the relative expression level of the RNA for each of these genes has been assayed for each of the patients. And then we use computational techniques to cluster the patients that are similar to each other. And you can see across the top that there's different groups or buckets of patients with different gene expression profiles. So we've got the light blue, dark blue, green, and red populations. So the red that Kaumudi is highlighting is really showing these patients have really strong expression of those red genes up top, and then really low expression of the green genes below. And so this is a specific group of patients. And if we do gene expression profiling, we can understand which group of patients your tumor is most similar to. We can look at historical data and understand which therapies are then most relevant or not relevant for that particular set of patients. So this is, again, as Kaumudi has said, can be expensive to do. It can be worthwhile, can be helpful. So it's something you want to talk to your doctor about, depending on what type of cancer you have, what stage in the journey you're in, and how much is known in your particular cancer type about gene expression profiling.
Absolutely. So we went through DNA-based testing. We looked at a little bit about RNA-based testing. And now let's look at the most common way in which protein-based testing is carried out on this tissue. So the most common way in which protein expression-based tests are done is through a process called immunohistochemistry, where the tumor biopsy tissue is taken and then is stained for the protein of interest. So here you're seeing with this arrow the protein expression on the cell surface. You see pieces of tissue from patient tumor biopsy samples, a case where there's really not the presence of that protein. In some cases, there may be low amounts or medium amounts or really, really high amounts of the protein expressed. And this protein expression then gives us clues about how the tumor is behaving, what sorts of treatments could be applicable, maybe promising in that case, and also sometimes prognostically how we expect the tumor to behave moving forward. And some of the examples here are the newest PD-L1 testing, which is looking at the expression of a protein, which is going to provide some information about whether these new immune checkpoint inhibitors are going to be effective or not. It gives us one piece of the puzzle. HER2 testing, which has been around for quite some time, as well as ER/PR hormone testing in breast cancer setting. BCL2, SST2A. There's actually several examples more. And more there are clinical trials which also recommend this sort of testing as a pre-screening process as part of their pre-screening for patients. So that's something to keep an eye out for. Another important point that comes up is regarding cost. And Adrienne, I know you had a little bit to say about that today, and so I'm going to hand it over to you.
Thanks, Kaumudi. I know here at Cancer Commons, you and I have both worked with multiple patients that have run into incredibly frustrating situations getting the proper biomarker testing paid for through their insurance companies. And we wanted to provide some reassurance and some guidance at the end of this presentation to remind everyone, and if you can just pull up the, yep, there we go, that most testing companies or laboratories will offer financial assistance based on need. So if your insurance, first of all, have the conversation with your provider about which tests are needed and which tests may be exploratory, what will or will not be paid for by insurance. And if you do get testing and the cost doesn't seem right, to inquire with the testing company themselves. Most of them will have a webpage just like you're showing here, where you can fill out for financial assistance. And then you can always reach out to organizations like us for support to help understand the process, understand if you've made the right choices in your testing journey, what additional tests may be recommended at this time, and how they can be used to help understand your prognosis and treatment options.
Absolutely. If you walk away with anything at all today, we'd like you to remember that we're here with you on this journey. And so even though some of the medical jargon, all these terms can be confusing and intimidating when you have to learn them all at the same time, we both consider it our passion to communicate this sort of information with people and walk with them in that journey. So
exactly, Kaumudi, and our nurse navigators and all the scientists here, we love doing this. We're very passionate about it, and we care about addressing each individual or caregiver for their very unique situation. Like Kaumudi, we had a registrant this week that on her intake form wrote in all capital letters, "So much information, so little room." And I thought that was exactly emblematic of what we're all experiencing, and you and I included, as we work with family members and friends going through their cancer journeys. And so we're really empathetic and understanding of how much information there is and how difficult it is to process, and we're here to help.
Yeah, absolutely. And with that, we'd like to open the floor for any questions you may have. Thank you very much for attending. Thank you very much for spending your time with us today.
Thank you, Adrienne and Kaumudi, for that great presentation on cancer biomarkers. And thanks to those of you who submitted your questions early. We've got some great questions here, so let's turn the time back over to our scientists.
So it looks like we have a question. Okay. It says, what are liquid biopsies, cfDNA, and ctDNA? Do you want to take this one, Adrienne?
Yeah, Kaumudi, this is a great question. Thank you for whoever asked this question. This is really timely and is going to be a big player in the biomarker field in the next few years. So this is something we didn't really dive into today. This technology is really emerging and is now much more accessible, but you're going to hear more and more about these liquid biopsies. So what are they? Well, we talked a lot about solid tumor biopsies, right? You have to take a bit of the solid tumor and then take some of that and send it to a lab to get sequenced. The liquid biopsy, as the name suggests, comes from some sort of liquid, most often blood, or there can be other fluids, bodily fluids that can be sampled relative to the type of tumor and cancer that is present. So there's a few other words here, acronyms here to learn. So there's cfDNA, which is cell-free DNA. And that's kind of the free-floating DNA that is shed into the bloodstream by dead cells. And then there's ctDNA, which is circulating tumor DNA. And so that is the free-floating DNA shed into the bloodstream by circulating tumor cells, or CTCs. So this liquid biopsy testing, there's some huge advantages, right? You can take these serially over time. It's a simple blood draw. You can look at changes in tumor dynamics and things like clonality, or which mutations are becoming more common in the tumor, or which resistance mutations are emerging. So there's a lot you can learn. The major limitation right now is that these tests aren't as sensitive, right? These are small fragments of DNA in your blood, so they're difficult to detect. So there's a lot of false negatives, which means if the mutation is not present in the liquid biopsy, we can't be really sure it's not there. So we have to take everything with a kind of a grain of salt. But these technologies are really active in the research world and being well studied. So this is a very exciting new type of biomarker testing that's going to become more and more common in the next few years. So thank you for a great question. I see another question here, which is, what is MRD testing? Would you like to take that one, Kaumudi?
Sure. MRD stands for minimal residual disease. And this is the sort of testing that becomes important in a few different scenarios. So for example, when there's something called NED, which is no evidence of disease, that's a really, really good news. There's no evidence of disease as seen by any scans, but you sort of still want to be sure that, and you want to have some ongoing monitoring and surveillance to understand whether there are any hidden cells that are floating around in the circulation or left behind, and that can be termed as minimal residual disease, and that's when MRD testing would be something you might want to consider if that's a possibility. Another scenario is where perhaps there's what is called a borderline resectable tumor, and successful surgery is performed. However, the tumor is at the margins, so just realistically, it's not possible to remove every single tumor cell from there, and there is a little bit left behind, and that is termed as minimal residual disease, and you want to monitor that over time, and that's when, again, MRD testing becomes important. Currently, as the field stands, MRD testing is most commonly used, or is used in the case of blood cancers, but it's still being tested in the case of other solid tumors. But this is something you want to definitely talk to your oncology team about, your oncologist about, whether it's a possibility for you or not. So yeah, another great question. Looks like we have another question. And this one is, what is HLA testing?
I can take that one, Kaumudi. So HLA testing, HLA stands for human leukocyte antigens. And so these are specialized proteins that are present on the surface of all cells except red blood cells. And we inherit the HLA genes that dictate which HLA antigens are presented on our cells. So you may be familiar with this from things like Be the Match, the bone marrow registry, or if anyone you know has had a transplant. So these HLA antigens are primarily used to match transplant donors and recipients. And that's a critical step in this matching process. And so the HLA testing identifies which antigens you have. And in the cancer world, it's relevant as some of the newer cancer vaccines, which are really showing some promise, they can be specific for certain HLA types. And so you need this HLA testing done to determine if you would qualify to be included in certain cancer clinical trials for cancer vaccines. So hopefully that helps explain that a little bit. Okay, Kaumudi, we have time for one last question. So I'm looking here. This is a really helpful question to clarify. What is the difference between serum tumor markers and cancer biomarkers?
That is a really good question. So let me sort of tackle this one. Serum tumor markers. First, let's look at that. Serum tumor markers are something that are typically looked at the time of, in order to diagnose or monitored throughout the course of the disease. Some common serum tumor markers include CA19-9 or carcinoembryonic antigen, which is CEA levels that are monitored over time. And many times people ask us saying, okay, we've already had this sort of testing done, so what is this other biomolecular or biomarker testing that you're talking about? So it's important to understand this difference. The serum tumor markers are important, but they really don't give the complete picture. So they're an important sort of testing to have in order to monitor the cancer, or also at the time of diagnosis, but they don't really provide information in depth about how the tumor cells may be behaving and what sorts of treatment options might be possibilities to consider. And so the kind of biomarker testing we talked about today is a more comprehensive look at the tumor cells. So whenever it's possible to do this sort of testing on biopsy sample or on liquid biopsies, as we talked about, it is extremely helpful to get this biomolecular testing or biomarker testing done in addition to the serum tumor markers that are routinely looked at for some cancers. So,
That's really helpful, Kaumudi. So we're running out of time here today, but we have some other great questions that we will be sure to follow up with either us or our navigator team here. We'll make sure to reach out to everyone else that asked questions. And I just want to thank everyone for being with us today.
absolutely, absolutely. Just remember again that we're here for you. Like Adrienne mentioned, our navigators, all of us scientists, we're here to answer your questions. So feel free to chime in with questions as they come up, ones that we didn't get to today. So thank you, everyone.
Thank you.
Thank you, Kaumudi and Adrienne, for a wonderful and informative presentation. And thank you all who attended. We will share the recording of this presentation with everyone who registered, and we'll be posting it to Cancer Commons website and YouTube channel. Please send any additional questions you have to education@cancercommons.org. If you or a loved one need assistance managing advanced cancer, Cancer Commons is here to help. Contact us via our website at cancercommons.org. And finally, Cancer Commons is extremely grateful to the Looney family for sponsoring this series. Our next webinar will be held November 9th and will focus on tools, strategy, and knowledge you need to take your case management to the next level. We look forward to having you join us.